Hello wonderful person, this is Anton, and today we're going to be discussing a somewhat intriguing discovery in the field of biology. And specifically, as the title suggests, we're going to be discussing the idea of cloning. Something that captured our imagination many times. For example, the idea of bringing back some kind of an extinct animal, like a mammoth or even a dinosaur, or even a more sci-fi type of an idea involving some kind of a biological immortality. Except that when it comes to that second idea, I have some bad news. Based on this radical 20-year study recently published in the Nature Communications, it basically suggests that for people, and specifically for mammals, cloning might not be as efficient and seems to have a tendency to eventually hit a genetic dead end. In other words, it does not seem to work indefinitely. But here we obviously have to explore this idea a little bit more, because not all of us are going to be on the same page when it comes to biology. And to understand what this means, let's first talk about how cloning works, and I guess what this means in general. And then discuss this Japanese study that basically studied 57 different generations in order to find out how this affects mice. And while University of Utah has this incredibly useful page that basically tells us a little bit more about the history of cloning, starting from the first demonstration in 1885 to some of the first cell control, first nuclear transfer, and of course the creation of some of the first cloned mammals, such as the famous sheep Dolly. But normally, when we talk about cloning today, cell biologists are actually only referring to one specific technique. It's the process referred to as SCNT, somatic cell nuclear transfer, a lab technique that creates a cloned embryo by transferring the nucleus of a somatic cell into an egg that does not contain its own nucleus. And so in some sense, it can basically be summarized this way. But in nature, cloning is actually extremely common. As a matter of fact, this right here, the aspen trees we discussed many times before, is a perfect example of a natural clone. And even plants like potatoes and bananas usually clone themselves pretty often. But it's just not as common in animals. And though some lower animals, like for example planaria or even certain lizards, can reproduce without a mate, this is a process referred to as parthenogenesis that's actually even been observed in sharks. For more complex animals, and specifically for mammals, things are just a little bit different. And that's because naturally our bodies are not meant to reproduce themselves through cloning. But artificially it is possible. And so here scientists usually take an egg cell, remove its nucleus, the part that usually contains the DNA, and then they take a nucleus from a different cell, or even from a different animal, and insert this into this empty egg. And then with just a little bit of a jump start, which usually involves a little bit of an electric jolt or certain chemicals, this new egg begins to divide as if it was actually a natural embryo. And so in mammals, the first demonstration was in 1996 with Dolly the sheep. And since then, scientists have cloned pretty much everything from various types of pets to even endangered animals like the Przewalski's horses. And obviously in some of the previous videos, we've even discussed the attempts to revive the extinct mammoths or direwolves. Though, as you might have learned from those videos, those were just basically a kind of a hype strategy. For example, for those direwolves, they were basically just your regular wolves cloned to contain certain genes. But because in the last 30 years technology has advanced quite a lot, cloning animals has become much more successful than before. Though there was always this one important question. Can we actually do this indefinitely? And could a typical mammalian species survive indefinitely by basically being cloned over and over and over again? In other words, can you actually create, for example, a copy of yourself that lives on for generations and generations to come? Yeah, we're talking about some dystopia stuff here. But still an intriguing question. And so to find out, the scientists from the University of Yamanashi began a massive experiment back in 2005. And they simply started with one female mouse and then just cloned her. So basically they produced the clone of the mouse. Then when the clone grew up, they took its cell and did the same thing again and again and again. In other words, they kept raising mice and kept cloning them instead of allowing them to reproduce naturally. And so starting with the donor mouse, they had the G1 clone, G2 clone, and so on. This was a type of a serial cloning that lasted for almost 2 decades. And at first, things looked great. For the first 25 generations, all of the mice were healthy, had relatively normal lifespans, which for mice is approximately 2 years, and overall the success of this procedure was actually improving. which is what you see in this initial graph, with researchers even at some point assuming that it might be possible to clone animals forever. But then the success rate suddenly began to drop, and by the 57th generation, the birth rate was only about 0.6%, which was very, very low. And then they reached the 58th generation. Every single mouse born from this generation unfortunately died after just one day. And so after 20 years, and after approximately 1200 mice used in the experiment, all of which actually lived relatively comfortable lives, this lineage hit a complete dead end. They could no longer produce any kind of a viable offspring, and the mice were just way too sick. And so the obvious question is, why? What exactly happened here? And the researchers here found that mice had accumulated a massive amount of genetic damage. And that's because every time there's a cell division, it actually has a tiny chance for a mutation, a mistake in the genetic code that just sometimes happens during division. And in normal sexual reproduction, these mistakes are very often filtered out or are basically corrected through the mixing of DNA from two different parents. Yet when you clone something, these errors just keep piling up and eventually become a very large part of the entire DNA, with a study discovering that by the 57th generation, the frequency of these dangerous mutations had nearly doubled. And here they even saw large structural changes in the DNA, including the loss of the X chromosome and the translocation where pieces of the chromosome broke off and attached to another. And as you can imagine, losing an entire X chromosome is kind of dangerous. And by itself, this is a very important experimental confirmation for a biological hypothesis referred to as the Muller's ratchet. The idea that in asexual lineages, or lineages involving clones, a lot of harmful mutations eventually accumulate, kind of like a ratchet turning in one direction. And eventually the genetic burden becomes so heavy that it leads to a kind of a mutational meltdown and even extinction. As a matter of fact, this idea has previously been used to explain why, potentially, Neanderthals went extinct. In other words, our cousins might have gone extinct because their genes were just not good enough. They were interbreeding too much and eventually accumulated way too many genetic problems, with the entire species basically disappearing. And something extremely similar we now know most likely happened to some of the last mammoths. The woolly mammoths that survived until approximately 4,000 years ago in the regions of northern Siberia on one of the icy islands. And so here, because the population was so extremely low, and because there was constant interbreeding, researchers discovered that because of the genetic problems, these mammoths eventually also reached an extremely similar conclusion. Their offspring basically could no longer survive. And so this experiment is a very important confirmation of many of these different assumptions about cloning and of course the importance of sexual reproduction. Or at least for mammals, because the next question here is, okay, so why can a potato and a banana do it, but why not us? Or basically, why can you clone a potato for centuries, but these mice seems to have failed after just 58 generations? And the answer here seems to be epigenetics. And that's because in a mammal, a skin cell nucleus actually knows it's a skin cell because certain genes are turned on and others are turned off. And so when scientists put that nucleus into an egg, they first have to reprogram the cell back into its embryonic state. And this process can be extremely difficult and can lead to abnormalities, including one that seems to be very common, enlarged placentas, something that seems to happen in a lot of these cloned animals. And while researchers can use certain chemicals to help in this reprogramming, the study shows us that even using perfect techniques, we just cannot fix certain things and certain underlying mutations. In other words, it just does not seem to work perfectly all the time. But here there was also a very important discovery in regards to how damaging this is long term, because apparently the damage was not necessarily permanent. As in, when the researchers took one of the females from the 50th and the 55th generations and then mated them with normal mice using sexual reproduction, something amazing ended up happening. At first, the babies were kind of small and they still had the same placenta issues and some of the problems, but not all of the problems, of the clone. But the grandkids were actually completely normal. In other words, all of the genetic damage was completely reversed in just 2 generations of sexual reproduction, which basically suggests that sexual reproduction is a kind of a powerful reset button that seems to eliminate many of the genetic anomalies that may have been caused by cloning. And so here it reaffirms that, at least for mammals, this seems to be an extremely important evolutionary necessity for long-term genetic health and of course survival. And naturally, this research also has quite a few implications on some of the studies and even some of the propositions by various biotechnology startups that we discussed previously. For example, that idea of de-extinction. So basically, startups trying to bring back the woolly mammoth or the Tasmanian tiger may definitely struggle long term, because cloning a single individual is just not going to be enough to save the species. Here this confirms you need to have a diverse population in order to avoid the accumulation of these mutations. At the same time, this also shows us that the conservation strategies used in the endangered animals, like certain types of horses, can actually work, assuming that we introduce the clones back into the original population in order to have them mate naturally. In other words, clones and not clones will definitely produce healthy population. And last but not least, the idea of pet cloning. So basically people paying thousands of dollars to essentially have their cat become undead. Oh wait, that's how the pet cemetery starts. Anyway, so cloning cats and dogs seems to be okay, unless you start doing this like 50 times. And so don't expect to bring back your pet generation after generation, because eventually they'll just become too sick. With the main point being that cloning, though a very powerful tool, is definitely not a substitute for the natural order, at least when it comes to mammals. And as this 20-year experiment shows us, even the most advanced biotechnology, unfortunately right now, cannot break the laws of evolution. The genetic shuffling used by sexual reproduction seems to be extremely important for keeping the species healthy. And so, anyway, on that note, we'll definitely discuss something very similar very soon. Until then, thank you for watching, subscribe, come back tomorrow to learn something else, support the channel on Patreon where you can find additional videos without any ads and can DM me directly, or by joining the channel membership that grants you early access. You can also support this channel by buying the Wonderful Person t-shirt in the description below. Stay wonderful, I'll see you tomorrow, and as always, bye bye.